Good morning. Today we are going to talk about an article with the title The Impact of High Fat Diet on Mitochondrial Function, Free Radical Production and Nitrosative Stress in Salivary Glands of Wistar rats. This article was published in the journal Oxidative Medicine and Cellular Longevity with an impact factor of 7.3 in the year 2019. So the title says High Impact of High Fat Diet, which means they are on the Wistar rats. So they are feeding high fat diet to Wistar rats, and they are just studying the mitochondrial function, free radical production, nitrosative stress. What is the mitochondrial function? So what is mitochondria? Mitochondria, we know it is the powerhouse of the cell. So it has an outer membrane and inner membrane, and the intermembrane space is there. There is a stray and there is mitochondrial DNA with ribosome. So this is the structure of a mitochondria, and the functions of mitochondria: it produces ATP, it does this mitochondrial respiration which produces ATP, regulation of immunity, calcium balance, cell death and renewal, that is autophagy, and stem cell regulation. These are the functions of mitochondria. So they are studying the mitochondrial function here. Free radical production. What is free radicals? What are free radicals? Free radicals are nothing but atoms which are ready to accept electrons. So these are free radicals, and the atoms which are ready to donate are antioxidants. So this is the free radical, and there are two types of free radicals. We have oxygen, reactive oxygen species as free radicals. We have reactive nitrogen species also as free radicals. So what does this free radical do? It will damage the DNA. And how the free radicals are formed? Either in inflammation during the metabolism, that is the mitochondrial activity, smoking, ionizing radiation, air pollution, and the UV light. These all produces the free radicals. Our body has antioxidants which counteract this free radicals and make a balance. But when this balance is broken, there is a lot of DNA damage. So when we talk about the reactive oxygen species, there are radicals and non-radicals. Here we are going to talk about only the free radicals. Radicals are nothing but freely available ions which are ready to react. So in the oxygen group, we have the superoxide, hydroxyl, peroxyl, alkoxyl, hydroperoxyl. All these things are ready to accept electrons. And in nitrogen species, we have nitric oxide and nitrogen dioxide. So what does these free radicals do in the salivary gland? Normally in salivary glands, the carotid, submandibular, subliminal glands have high secretory activity. They are always in the production mode. So what does the mitochondria do? They provide ATP needed for the ion transport and protein synthesis. So the mitochondria is constantly working here. This produces an oxidative stress, and free radical also is increasing. But during the high activity, mitochondria generates reactive oxygen species, and in salivary glands, the excess ROS, that is oxidative nitrosative stress, damages the acinar and the ductal cells. The antioxidant enzymes such as superoxide dismutase, catalases, and glutathione peroxidase, they are present normally, which can balance the oxidative stress which is produced during this mitochondrial normal physiological activity. So this is how the mitochondrial function is related to the salivary glands. Now they are going to test a rat which is fed with high fat diet, and the difference in the salivary glands function here. So the novelty is, it is already proven that oxidative stress and salivary gland dysfunction in the course of the insulin resistance induced by high fat diet, and the role of salivary glands in maintaining the redox homeostasis. And it is important to learn about the mechanisms linked to disrupting this balance. There have been no studies investigating the impact of high fat diet induced insulin resistance on mitochondrial function of the salivary glands of rats. And there are also no available studies analyzing the changes in the activity of other enzymatic intracellular sources of ROS and nitrosative stress in these glands. So the aim of the study was to investigate the effect of insulin resistance induced with high fat diet on mitochondrial respiratory system, the activity of pro-oxidant enzymes, ROS production, and nitrosative stress in salivary glands of insulin resistant rats. So here they used male Wistar rats. These are the male Wistar rats, which is a strain of laboratory rats characterized by thick long ears, long body, wide head, and short tail. This is the Wistar rat which they used. So the rats were randomly divided into two equally numbered groups, that is 10 in each group, that is the control group and the high fat diet. So how did they plan the study? For 8 consecutive weeks, The rats control group was fed with a normal diet, whereas the high fat group was fed with the diet with more amount of high fat. Then after 8 weeks, all the rats were fasted for 12 hours and they were anesthetized, and all these tests were done. The BMI was calculated, the fasting tail blood glucose, the non-stimulated as well as stimulated salivary flow was calculated, blood samples were taken, and salivary glands were retrieved. Both parotid and the submandibular were taken. So the non-stimulated saliva was measured for 15 minutes. How did they use? They used the pre-weighed cotton balls, inserted into the oral cavity underneath the tongue. So this is how they measured the non-stimulatory one. In the stimulatory saliva production, they used pilocarpine, which was injected into the intraperitoneally, and the salivary secretion was examined for 5 minutes. Pilocarpine is a cholinergic agonist. It works by stimulating the muscarinic receptors. I said parasympathetic nervous system stimulates the saliva, so it increases the secretions from exocrine glands like salivary glands and tear glands. So this is how they studied the stimulated and unstimulated salivary secretion. And later, after retrieving the salivary glands, they homogenized on the eyes with the glass tissue homogenizer, and we need to study the mitochondria. So they isolated the mitochondria as well. And the ROS production. As I said, the ROS is produced by the mitochondria with the help of all these enzymes, and which causes the insulin resistant mitochondrial dysfunction, cancer. All these things are caused by these reactive oxygen species. So the activity of in this study they analyzed the NADPH oxidase function, activity of xanthine oxidase, and ROS production was estimated spectroplurometrically. So in this pathway they studied this and the ROS which was produced. And in the they studied the mitochondrial activity. So the mitochondrial activity they have a complex 1, 2, 3 and 4. There are different steps for the respiratory thing which is happening inside the mitochondria. So in this activity they studied the complex 1, complex 2 plus 3 and activity of the cytochrome oxidase enzyme, which was also studied. And the production of mitochondrial hydrogen peroxide was assayed using Amplex red horseradish peroxidase method. ADP-ATP ratio was also measured. And they also studied the glutathione metabolism. So glutathione is an organic compound made of amino acid glutamate, cysteine and glycine. It is an antioxidant. So ROS is the free radical which is produced. Glutathione is a substance which counteracts the ROS. So they studied the counteracting ability of glutathione also in this study. So this is how the glutathione is produced and it counteracts the ROS. So the level of total glutathione was assayed and the level of oxidized glutathione, this one was measured, and the redox state was calculated using the formula GSH square by GSSH. And the activity of glutathione peroxidase was assayed calorimetrically, and the glutathione reductase was also estimated. So all the this pathway, in this pathway they have measured almost all these steps to assess the glutathione activity. And next they measured the oxidative and the nitrosative stress. So the level of monoaldehyde was measured colorimetrically, nitric oxide was measured, and the level of peroxynitrate was analyzed colorimetrically based on the peroxynitrate mediated nitration. And nitrotyrosine was also evaluated using ELISA. Inflammation apoptosis was also studied using the level of interleukin for inflammation and the activity of caspase 3 for apoptosis. So this thing they had measured, and the protein assay. The content of total protein was also used by BCA method with bovine serum albumin as a standard. And they conducted a statistical analysis using graph pair prism. They checked the normality and for test, and unpaired student t-test and peer correlation method was implemented. Sample size was based on the pilot study conducted earlier. The statistical power of the test was assumed to be equal to 0.9. So in the results, they compared the submandibular glands, this is the parotid glands, in which the controlled rats showed the xanthine oxidase and caspase 3 activities, GSH and NO concentration and redox ratio significantly higher in the parotid glands. And the controlled rats demonstrated significantly higher complex 1 and GSH concentrations in the submandibular glands compared to the parotid glands. In the high fat diet rats, the activities of NOx, xanthine oxidase, CS and CAS3, all these things were significantly increased in the parotid glands. And the hydrogen peroxide production, GSH concentration and redox ratio were higher in the submandibular gland compared to the parotid glands. So the correlation which they studied showed a negative correlation between the mitochondrial hydrogen peroxide production and complex 1 activity in the parotid and submandibular glands of HF3 rats. A positive correlation was noted between the DCFHD concentration and NOx activity, while negative relationship observed between peroxynitrate concentration and complex 1 activity in the parotid glands of HF3 rats. So in conclusion, the 8-week intake of high-fat diet impact the mitochondrial function in both types of salivary glands by enhancing the ROS production, and the ROS concentration was higher in the mitochondria of submandibular glands. NOx activity, DCHFH production, protein nitration and lipid peroxidation were more pronounced in the parotid glands of the HF3 rats. So the limitations of this study were like the findings were in Wistar rats and which may not directly translate to human physiology. Only high-fat diet was tested and other dietary patterns were not explored, which focused only on salivary glands. The systemic and metabolic effects were not assessed, and it is a single duration fat content, which dose response was not studied. The possible bias if only one sex or age group. Here they use only the male Wistar rats, so that would be a bias here. And the functional outcomes, that is the salivary flow, oral health were not measured. The future scope of this study is to extend the study to human or clinical models of translation relevance and compare the effects of different diets, high sugar, mixed diets or protective diets, explore molecular pathways. These pathways can be observed, and we can test the interventions by using antioxidants, exercise and dietary modifications, correlate biochemical findings with the salivary flow composition and oral health, and conduct a longitudinal study to compare early versus late diet induced changes, and analyze the sex and age group specific responses on high-fat diet, and investigate the systemic organs crosstalk, that is liver, adipose tissue, pancreas, all these crosstalk that can be studied. And this study followed the ARIVE guidelines, which almost followed all the criteria of these studies. The blinding was not done, randomization was not properly mentioned. Inclusion exclusion criteria, they included only the male Wistar rats, other than that they did not mention any differences here. Otherwise, all the protocol registration was there, and the declaration of interest, data access was also mentioned.